Okay. Uh, Hello, welcome everybody to Biomarkers. Our speaker for today is John Wendt from the Oklahoma State University, USA. So John is here to talk to us about, reconstructing past herbivore ecology with bones and biomarkers. Um, so the format for today's seminar is we have a welcome announcement, which is currently going on, and then we have our talk for approximately 30 minutes, followed by a moderated Q&A. And then we have tea time, which John has very kindly agreed to stay back for. So if you have any questions, please send it to us via chat to questions@RadioWorks. Our questions host for today is Nina. And a bit of housekeeping before we actually start. So at PaleoWorks, we value the participation of all folks who are interested in all kinds of value sciences. And we would request you to abide by our code of conduct for today's seminar. Please mute yourself for the entire duration of the talk. And you can send your questions to the questions host at PaleoWorks, or you can use the raise hand function. Should you face any technical issues, please reach out to the questions host. And we have currently the closed captions enabled into Zoom. So you can use the CC button to show or hide them. And we would request you to nominate an outstanding ECR if you know any. And here's the link. So the link will be sent in the chat box. And we would also request weekly feedback form for you all to fill in for our demographic info. And now, so a bit about our speaker for today. So John has done his BAs from the Colorado State University, followed by a PhD in the Montana State University. And he's currently a postdoctoral research fellow at the Oklahoma State University and Climate Hub fellow with the USDA Southern Plains Climate Hub, USA. So over to you, John. Have a great talk.
All right, thank you. And thanks everybody for joining. Let me just hide this here. Okay, so I'm excited to talk about some of my work on herbivore ecology using bones and biomarkers today. So let's just dive right in. First, I'll talk about approaches to reconstructing past herbivores, following kind of a broader scale look down to a more smaller, shorter term and more local look. So the first two parts here are focused on bones of bison, and the third part is focusing on molecular biomarkers in Yellowstone National Park. And then I'll also, give my thoughts on some future directions to some of this work. So just quickly about me, as it was mentioned, I have a background in through Colorado State University. I went through Montana State University for my PhD, and now I'm at Oklahoma State University. My background is actually in rangeland ecology as an undergrad, and with a lot of work experience too. I traveled around surveying rangelands, studying abroad in Mongolia, just getting a really good idea for, or just with an interest in kind of spatial variability of rangelands. But I had these nagging questions about, well, were these systems always like they are today, or how have they changed? So that motivated me to look into paleoecology, which I pursued for my PhD. So for that, I had a lot of fun in mud, coring lake sediments with the lab here, and coring wetlands. For those of you who don't know, the lake sediment work in paleoecology, and this is a wetland core actually, you can extract lots of information from the past just from looking at the mud. You can look at tephra to identify past volcanic eruptions and constrain the age of sediments. You can look at pollen to reconstruct past vegetation. Charcoal can give you an understanding of past fire activity, fire variability. And biomarker molecules can also be used to infer things about past organisms. So why do paleoecology? Why focus on rangelands with paleoecology? From a management perspective, current paradigms focus on maintenance of ecological states and often refer to some past conditions, but in a world that's rapidly changing, it'll become increasingly difficult to maintain static ecological states. So paleoecology can tell us how rangeland systems respond to rapid environmental change. So two important components of rangeland systems are large herbivores and fire. They influence the structure and dominance of plants. They influence fuel loads and fire patterns, the evolution of plant traits, and nutrient cycling and other things. So they're important to understand in order to understand how rangeland ecosystems work. But rangeland long-term disturbance patterns in rangeland ecosystems are understudied and poorly understood. Despite covering nearly half of Earth's terrestrial surface, grasslands, savannas, shrublands, and deserts are not well represented in paleoecological reconstructions. For example, they only make up 16% of sedimentary charcoal databases, which are indicated by the red points on this map here. And herbivores are even less well understood. There's just far less data there are far fewer long-term continuous reconstructions of different dimensions of herbivores. So to tackle that challenge for my PhD work, I looked at variability of herbivores across different scales, focused on North America, so across different temporal and spatial scales. So for this first part of the talk, I'll talk about large-scale climatic drivers of bison abundance and distribution since the last glacial maximum in North America. Then I'll discuss using some of the data that we developed for the chapter 2 here. I'm going to relate bison abundance to past fire patterns in mid-continent North America during the Holocene. And then we'll focus on a lake in the northern range of Yellowstone National Park and how I applied biomarkers to reconstruct past herbivore impacts and community composition. So with this first bit, we'll talk about, we asked, how has climate change influenced the distribution and abundance patterns of bison since the last glacial maximum 20,000 years ago? So this started and was inspired by work. Well, just this, I looked at kind of the bison observations in a database, in the Neotoma database, and just kind of threw them all together. This is in the early days of COVID when I wasn't allowed to go into the lab. And it was just really striking to me how dynamic bison distribution was over time. People might have this idea that bison were just abundant in the Great Plains and they're a Great Plains species, but this data suggested that the story might be a little bit more complicated. Also, you can see on the bottom here, the inferred change in bison body size from bones that other folks have worked on. So that's also a really striking change, right? Bison body mass was reduced by about 50% during this time period. So when we're reconstructing past herbivores, there are a few dimensions to consider, right? There's identity. So at the community level, how is the herbivore community composed? What species make up it and how dominant are they? Quantity, so in terms of relative abundance, population numbers, or density of herbivores. And then ecological impact, so how are herbivores influencing vegetation, soils, erosion, water resources, etc. So proxies for paleo herbivores, we have a few options. The first here, palynological indicators. So these would be microscopic indicators of herbivores or herbivory. So take, for example, the spores of fungi that grow on dung. And these proxies have their strengths and weaknesses. So you can't really get at identity with these spores because this is a separate species that might grow on any animal dung or herbivore dung. And then quantity, there are some questions about its sensitivity, especially at low herbivore densities. It's also a biological proxy, so it has its own ecological sensitivities that might filter it and might be the processes that filter it might be unrelated to the actual number of herbivores. So also we can consider fossils, so preserved animal bone and other tissues. So those can provide an indication of identity and potentially quantity, depending on the context and the scale that the fossil record is being analyzed at. Then these bottom two, I think, are really exciting. Ancient DNA and molecular biomarkers, both are molecular approaches. Fossil DNA, in this context, I'm referring to DNA in sedimentary records, so like environmental DNA in sediments. That can tell us about identity of past herbivores, and then quantity maybe with replicates. You might be able to get at pseudo abundance. Whereas with molecular biomarkers, I think they're pretty exciting because they can constrain identity and also I think they can provide an indication of abundance or quantity. So first we'll discuss, for the first two bits here, we'll talk about approaches with bones. So this is back to that data set of bones, of fossil bison bones over the past 20,000 years. And this is just the frequency distribution plotted over time with a correction for taxonomic loss. So the raw distribution is much more biased towards recent deposits, but if we correct for that bias, then this is an attempt to have comparable abundant frequencies that are comparable across time. So there are a few interesting things about this distribution here. One that I'll point out is this period where megafauna in North America went extinct, where every animal larger than a bison in North America went extinct during this window. Bison, there's some considerable variability, but if you look at this window in total, it looks like bison may have become more abundant. They at least, and maybe even experienced some ecological release. So I think that's worth looking into more. So to understand bison distribution patterns post-glacial, we looked primarily at climate. We just looked at climate drivers in a species distribution modeling framework. We linked the bioclimatic variables to the observation data set and predicted the distribution of bison based on that. So this is primarily a climate envelope analysis. So, it's interesting. Bison are widely distributed, but most of the observations are in the Southwest during the last glacial maximum. Then by the Younger Dryas, bison are in some continuous band from Mexico all the way to Alaska, basically along the spine of the continent through the Rockies. And then it's not until the late Holocene where we really see this distribution of bison, spread throughout the Great Plains into the Prairie Peninsula in the east. They're throughout the Intermountain West too. So this is what you might think of as a more historical bison distribution. So looking at some of the variables that predict bison distribution. So there's, on the right hand here, we have variable rank. So during the Holocene, temperature of winter, average winter temperature was the top predicting variable. And this blue band is the value of that variable within the predicted distribution of bison. So bison, this is showing that bison prefer, say, warmer environments than what was available to them in the North American continent, as indicated by the green here. So bison enjoyed warmer winters. here's some of the other variables. I won't get into them right now. And then precipitation. So another important variable in our analysis was precipitation of the coldest quarter. So again, a winter variable. Bison tended to be on this low end of precip. So perhaps bison were, just avoiding cold winters, avoiding deep snows in the winter. So if you're interested in exploring this data set of bison fossils yourself, this is taken from Canadian Archaeological Radiocarbon Database and Neotoma Paleoecology Database, but we applied some stricter filters to get just higher quality observations. So if you are interested in that, feel free to follow the link. So just a quick summary. In terms of geographic distribution, we find that bison shifted from the southwest to the mid-continents over this 20,000 year time period. Extirpations took place at the margins. Say you find bison in Southern California during the last glacial maximum, but not during the late Holocene so much. And in the late Holocene, bison really came into their more historical distribution throughout the Great Plains and the Intermountain West. And then abundance changes, there was regional patterning to the changes. It was highly variable and influenced likely by ecological interactions and climate. So potentially ecological release, potentially influences from predators at different points in time in different places, and climate change as well. So building on the bison observation data set, we then went on to this next effort to understand how consumer dominance and prevalence respond to spatial and temporal moisture gradients in Holocene North America. So this is motivated by some work in modern sub-Saharan Africa that I'll get into in a little bit, but it's basically looking at the relative distributions of fire and dominant herbivores being bison over time with, as climate changed during the Holocene. So there are these questions of, well, in Africa, we see dry open grasslands characterized by these vast herds of diverse herbivores. And then we have also these wetter mixed savannas that are kind of understood as fire dominated systems. So did this pattern also play out in North America historically? Did large herds of bison and other herbivores dominate in the dry open grasslands whereas fire was more important, where it was a bit wetter? And also another thing to consider is there are these perspectives and they seem to be in conflict, right? Where on the left panel, we have this notion of competing consumers where herbivory limits fire. So fuels are broken up by herbivores. There's less fuel in the landscape to burn because of all the consumption that herbivores are doing. Whereas on the other hand, fire can limit herbivory. So if something is burned, it cannot be grazed, right? So, and also fire has destructive effects potentially for herbivores. Whereas there's also this perspective, especially in certain regions of North America, where called pyrrhic herbivory, where fire drives herbivory. We find that in some places and sometimes herbivores will actually seek out recently burned places. And cows here are quite curious as to what's going on. They might go and graze the burned patch immediately after a burn. So looking at the patterns in sub-Saharan Africa, we see across the precipitation gradients, herbivores have this curve where they're most prevalent on this drier end of the moisture gradient, whereas fire is most prevalent a little bit in wetter systems. And there's some overlap in these distributions too, right? There are some places where there's virtually no fire or virtually no herbivores. But so here on this low end, where it's 200 millimeters mean annual precip, herbivores consume almost all consumed biomass, whereas fire consumes virtually none. So does this relationship extend to North America? That's what we predict that it would. And this is just a kind of abstracted, hypothesized relationship that based on that last curve, with herbivore dominance in drier systems, both fire and herbivores kind of co-dominant in more mesic systems, and then wetter systems dominated by fire. So to address this over long time periods, over the Holocene, we looked at all these different sites. Some have fire, some have pollen data, some have moisture availability as a supplementary variable for us. And then you can see the regions are kind of broken out into these different precip zones, where the northern grasslands are drier, the grassland forest transition is wetter, and they roughly correspond to these herbivore dominant and co-dominant kind of predicted zones. So a bit about the data. Biomass burning inferred from charcoal influx into lake sediments, bison abundance based on that previous data set where site frequency with the taphonomic correction applied. Moisture availability with multiple proxies to characterize droughts and trends in hydroclimate. And then woody plant dominance is inferred by the ratio of arboreal to non-arboreal taxa in sediment cores with pollen. And then also we looked at modern data with these variables. So some remote sensed fire products, some census-based data for livestock, and the world climb data set for moisture availability. So when we take all the data and we aggregate them by region, and then we standardize the data, we see these patterns over the past 10,000 years on the x-axis. You can see the northern grasslands on the left, the grassland forest transition zone on the right, so drier on the left, wetter on the right. So what do we see? We see both this throughout the entire period. You can see that bison were more abundant in the northern grasslands compared to the wetter region. We see that fire burned more biomass, or more burning took place in the wetter region compared to the drier region for much of the past 10,000 years. We see that the northern grasslands, as you might expect, were more open first compared to the grassland forest transition, which was had more woody plants. You also see that these patterns correspond somewhat to changes in hydroclimate, which is indicated on the top here with the blue bars or the blue lines. So, you know, regional patterns of herbivore abundance and biomass burning were influenced by regional hydroclimate, but they also tended to these regions are distinct in terms of disturbance and the causes of biomass turnover. So if we just take the real, you know, for each of these points here is a point in time. And if we just match grazer abundance with biomass burning for these two different regions, so the drier region on the bottom here and the wetter region on the left, upper left side here. So you can see with lighter lines and dots corresponding to older observations progressing towards more recent. So you can see that both these regions started out more similar and over time became more differentiated with respect to their dominance, the dominance of either herbivores or fire. So if we look at this across a moisture gradient, so we can infer moisture availability by just looking at that ratio of woody to non-woody pollen. So on the drier end, we see the bison observations. Bison were most abundant on the drier end again, compared to the wetter end, and then fire is most abundant in the wetter region. So this is just kind of our attempt to reconstruct those curves based on that we hypothesized based on Africa. And now this is what modern North America looks like today. Using the same units that they use for the Africa study, we can see that herbivory dominates biomass consumption across all of the moisture gradient in North America. Fire doesn't even really register. And we can also look at how this relationship maps spatially. So this is the modern data, and you can see throughout North America, the yellow indicates a greater proportion of dry matter consumed by livestock, whereas the purple zone is indicating near 100% biomass consumption by fire. So again, this is modern data. And there are a few regions where fire is still important, in the mountains of the West, in some rural areas with fire cultures, and also obviously in the boreal forests in the north. And then across the moisture gradient, this is the proportion of dry matter consumed. So at a continental scale, herbivores dominate biomass consumption at all moisture levels. It's not really even close. But if we apply the Africa model to North America as a kind of a guess as to how it was in the past, we see this, right? We see this differentiation between East and West, where the wetter regions are expected to have more fire, the drier regions are expected to have more herbivory. And then there are these zones with a mix where fire and herbivory could have been co-dominant consumers of biomass. So to conclude this section, historical distributions of fire and burning in mid-continent North America do resemble those of contemporary sub-Saharan Africa in interesting ways. Herbivory dominates biomass consumption today, while fire is now functionally absent from regions where it was once common. And so implications for managers, if you remove important disturbance processes that ecosystems are dependent on historically, you might have there might be some undesirable consequences if that change in disturbance persists, let's say. So managers would be well advised to consider the relative dominance of fire and herbivory in the system that they're managing. Okay, so now on to the biomarkers. So this biomarker study it originated with I was with my advisor Dave McWethy. We were driving out to the National Bison Range. This is before my program even officially started. We were just collecting some sediment cores, and he mentioned that he was working on some sites and at these sites he was looking at caprostanol. So that's a biomarker that has been used to infer past human presence in sedimentary records. So I got thinking, well, I have this rangeland background. I'm interested in herbivores. There doesn't seem to be a lot of attention. There's some, but not a lot of attention on herbivores in paleoecology. What if we applied these biomarkers to wild herbivores? People had applied them, and I'll talk about this in a little bit, to domestic herbivores, to archaeological settings, but and some people kind of dip their toes in the water with regard to wild systems or like semi-wild systems. But here we sought to apply biomarker approaches to lake sediments in northern Yellowstone National Park. So we set out to characterize decadal to millennial scale changes in herbivore use and community composition in the Yellowstone Northern Range. and to evaluate the environmental data to better understand how large herbivores in the northern range shaped, responded to, and interacted with their changing environments. So our strategy involved reconstructing long-term herbivore identity and abundance by measuring fecal steroid biomarkers in lake sediments. So these are some of the key compounds that we looked at. This is not all of them, but we looked at ethylcoprostanol, and coprostanol. These two are produced by enteric microbial modification of dietary steroids. So in the case of 24-ethylcoprostanol, it's a phytosterol that is being modified in the digestive tract. In the coprostanol case, it's cholesterol. So it's not found in it's not abundant in plant food. It's abundant in animals though. So it indicates either, say, like internal sloughing of the animal's biomass itself, or of a diet that consists of some animal inputs. And then lithocholic acid is a bile acid. There are other bile acids that we looked at, and bile acids are cool. They're synthesized by the liver. They're genetically kind of determined, so they're less influenced by diet. So they provide some pretty nice details sometimes into which animals were producing them. So a little bit about how these compounds are produced. So if we start in the middle, in the top middle here with the delta 5 sterols, so this is where cholesterol is. This is where the phytosterol that I mentioned earlier is. And there are two different pathways for these compounds to go. If they're reduced by microbes in the environments in an aerobic setting, they are transformed into 5-alpha stanols. Whereas if they take the other route and they're reduced in an anaerobic situation, often in an intestine, then that leads to 5-beta stanols. So those are the compounds that we're interested in. We're interested in the 5-beta stanols, and we're also interested in the bile acids, like I mentioned, which are synthesized in the liver, excreted into the digestive system, and then recirculated, but some of it leaks out into the environment. So some previous applications of biomarkers. So looking at quantity, here is a study looking at this black line here is a ratio of different steroids, including coprostanol. And then the bottom panel is our independent estimates from archaeological data to estimate population at Cahokia. So metropolis in Mississippi Bottomlands near St. Louis, present-day St. Louis. So what's important here, though, is just how the quantity of the biomarkers follows this independent population reconstruction. Okay, so it can likely tell us something about population change. Also, fecal steroids have been used to characterize source abundance, source identity. So this is an application in archaeological soils, looking at soil samples and dung samples, so comparing the two. So the purple samples are reindeer, the green samples are horse, dogs, and humans also, and the black dots here are soil samples. So this shows that the primary contributors to the fecal steroid signature of these soils was reindeer, and in one case it looked more like a horse. So you can learn something about identity of sources as well. So to introduce the system, this is the Yellowstone Northern Range, home to it's on the border of Montana and Wyoming. Part of the Northern Range is in Yellowstone National Park, but not all of it. And this boundary really is depicting the historical Northern Range. Bison today aren't really allowed to roam all the way down to Chico, down valley. So it's home to bison, it's home to elk, moose, mule deer, pronghorn, bighorn sheep. So a panoply of North America's iconic herbivores. And to understand our results, first we have to understand kind of the different phases of management in the system. So Yellowstone National Park and the Northern Range went through these three periods that we characterized. So beginning in the late 19th century, Yellowstone National Park is established, hunting is prohibited, predators are controlled, wolves are eliminated, and this leads to growing herbivore populations. And that is really the intent. It's to protect bison, to protect the elk herds. It's kind of a reaction to the slaughter of these animals elsewhere. So it's a preservation mindset. But concerns of overgrazing and resource degradation are grow. So then the park starts managing bison and elk populations. They start applying scientific management tech approaches, including aerial elk surveys, rangeland monitoring. And a turning point was the publication of what's known as the Leopold Report, which is kind of questions the orthodoxy or the wisdom of these heavy-handed management approaches. So that leads to the natural regulation era, where a adoption of the natural regulation policy leads to the end of intensive management. Bison migrations begin again. The animals' ranges expand. Predators recover. Wolves are reintroduced, and other predator populations recover. But during this period, there's still uncertainty. There's still uneasiness about how the park is handling its animal populations. In this report, which is a Natural Resource Council report, so pretty high status publication with lots of different authors, with lots of different expert input. They say that our lack of understanding about previous conditions and current dynamics makes it difficult to reject the hypothesis that ungulate populations will be naturally regulated without causing long-term damage to the greater Yellowstone ecosystem. So knowledge of characteristics and dynamics of past herbivore communities is considered crucial for understanding how these ecosystems function, and such knowledge would help define the historic range of variability, which could help define management targets. But the existing fossil record provides very little information regarding past ungulate abundance and ecological impacts. So there's a need for this kind of information that biomarkers can provide. And this is in my background picture, Buffalo Ford Lake in Yellowstone. So this is where it is geographically. So follow the red X, and it's this in a small catchment in the lower Lamar Valley, near the confluence of Slee Creek and the Lamar River. So I'll go back to that. If you've seen pictures of large herds of bison in a grassy river valley in the summer, you're looking at the Lamar River Valley most likely, at least if it's a Yellowstone picture. So we collected some dung samples from bison, elk, moose, mule deer, and pronghorn. We analyzed those dung samples for a suite of steroids, including the 5-beta stanols that I mentioned earlier, bile acids, and some others that provide some information but aren't quite as specific. And then also we characterized sediment from Buffalo Ford Lake. So we collected a core that was about 160 centimeters deep from the deepest portion of the lake, and quantified these compounds too. So I'll start here with just looking at the dung samples of the different species. We can see in this top right panel, most of the species kind of fall out into their own clusters, with some overlap between elk and bison. So what's interesting here, though, is how we go from, as we go left to right on this panel, we can see basically, as you go left to right, the animals go from browsing towards grazing. So as a proportion of diet, moose have limited grass intake and a lot of woody plants, whereas bison prefer grass. And then the animals in between follow along that gradient as well. So this is kind of suggestive of dietary and potentially anatomical, genetic, altogether influences on these fecal steroid distributions among wild animals, wild herbivores. So then we compared the fecal steroid signatures of these herbivores to the signatures we found in lake sediments. So just looking at the 5 beta stanols, so on this PCA on the left, you can see caprostanol, epicaprostanol, 24 ethyl caprostanol, and its epimer. So those with those 4 compounds, we can see that pronghorn, moose, bison, and elk mostly fall into their own separate clusters. And then if we plot these clusters out and add the lake sediment samples and their steroid profiles, we can see following the black dots here in the 3rd panel that the lake sediment most strongly resembles the fecal steroid signatures of elk and bison. And that's throughout the entire 2,000 plus year history of this sediment core. So this leads us to believe that grazers dominated throughout this period. There's no point at which any other of the other clusters are closer. So now let's look at abundance, and this is back to the kind of this historical record. We're just looking at the past 150 years or so. And so you can see that this blue line with the zoostanols, it has this dotted line here. It reflects the 2,000 year average. And then the solid line are the individual observations during this period. And then the gray lines are just the date uncertainty from the lead 210 dates that we have for this section of the core. So you can see there's a dramatic increase in the highest levels of the record took place about in the 1920s, when just as this preservation approach was giving way to population control, just as elk populations were at their maximum during this period. It's important also to note that both elk and bison during this time were being provided hay during the winter, and they were not migrating down valley. They were actually staying up close to the Buffalo Ford Lake area. The winter haying stopped. Bison still wintered in lower Lamar Valley for a few decades. And during this period, we also see another peak in the total zoostanol signature. So we actually think that with the cessation of winter haying, the bison shifted slightly down valley and basically congregated around our lake. And there are historical documents suggesting this. Then bison and elk were allowed to expand their ranges, and you see
Hi,
less of a
John.
correlation. Yes?
Yeah, this is just to let you know that we are at 40 minutes. We're over 40 minutes.
40? Okay, I'm almost done.
Yeah, sure.
I'll wrap up. So this is just to show that the correlation is between the biomass and the stanols is stronger during this population control era when animals were more spatially concentrated around the lake, and then that correlation became weaker as animals spread further out across the landscape. So just quickly to relate this to ecological impacts, we see willow decline in the early 20th century, grass decline as well. The grass recovered, the willow did not. This is related to this has implications for the trophic cascade hypothesis. If you're familiar with that, we can talk about that more in Q&A if you're interested. The lake system itself also seems to have been influenced by these herbivore changes. So increased sediment accumulation rates when the zoostanals are peaking. We can come back to this if there are questions, but this is the 2,000 year record compared to the fossil record and our paleo environmental proxies. And this is kind of our working model of how the fecal steroids get into the lake and what time of year they get into the lake. So in this system, animals congregate in valley bottoms during the winter. They seek greening vegetation in the spring. So they spread out across the landscape. The snow melts, the ice thaws, dung ends up in the lake sediments. And then by summer and fall, the animals are more broadly distributed across the landscape. So perhaps there's less of a summer and fall signature in this data set, in this data. So just to wrap this up, we found that fecal steroids in lake sediments can be used to reconstruct variability in species dominance, local herbivore use, and their environmental impacts. We found bison and elk dominance over the past 2,000 years. And the 20th century was really historically exceptional in several ways. So briefly, future directions. I'm really interested in continuing to work with these sedimentary biomarkers for wild herbivores to develop long-term records to compare with past drought, past fire, vegetation, soil dynamics. Also, there's work that needs to be done to establish quantitative relationships between these biomarkers and the processes that we think are influencing them. So thank you all for listening. You can find me at my website, or here's my email address. And I just want to acknowledge all the people that contributed to this work here. I'm not going to list them all, but my great thanks. And if we have time, I'd be happy to take some questions.
So thank you so much for giving that lovely talk. We have a couple of questions. So our first question is from Roy Plotnick. So the question is, the bison numbers post megafaunal extinction decline and then recover. A recent PNAS paper suggested a continental peak US indigenous population, approximately 1150 CE, which declined and stabilized by 1450 CE. Is there a possible correlation there?
Yeah, that's a great question. And that's something that I'm really interested in exploring in greater depth. So yeah, when we see this decline here at the end of the record, so this first dotted line here, this is the 1,000 year mark. So 150 CE, sorry, 1150 CE would be around, it would be a bit, let's see, I'm really bad transferring BP to CE. So, excuse me, it would be around here, yeah. So yeah, like I was saying, they also see this kind of steep drop off in their data as well. At first, we kind of like weren't sure what to make of this because, oh, is this just reduced interest in bioarchaeologists in dating these observations? A lot of our observations and this database is a radiocarbon database, so maybe there's some maybe just during this time period, archaeologists are just using other means of dating the bison. So that caused us to question, well, like, is this a real peak? Or is this just an artifact of research interest? And there is some overlap in the data between the PNAS paper that you're referencing and our data. We both use the same radiocarbon dates, at least. They have far more, and not all of theirs are bison dates. But yeah, they are pretty confident that what they see in that drop off is real. They don't think that it's an artifact from the declining researcher influence or just different methods of dating or decreased interest in more recent sites. So yes, I think there is a correlation. I think we'd have to look at this at a much finer scale and to really be able to say anything definitively right now, but it is certainly suggestive. When I look at the data more closely, just kind of anecdotally, I wonder about things like disease, right? So we know that disease affected Native American populations, and I wonder if any bovine diseases from introduced livestock may have contributed to this apparent decline in bison as well. That would be a little bit later, obviously, than the 1150 CE date that you mentioned here.
So our next question is an anonymous question. So the audience is very interested in the Shiny app that you mentioned. Could you provide a link for it? Apparently, the QR code went by a bit too fast.
Sorry about that.
Yeah. So the
Yeah,
Shiny app that you mentioned, the audience is very interested in that and would like for you to provide a link. So if you keep the QR code on the screen for a bit longer.
sure. There it is.
Yep.
If someone can copy, oh, let me see. Might be able to copy it into the chat too. Oh, sorry.
Great. Our next question is, do you know if there are any management plans to reintroduce fire consumption or otherwise make the ecosystem more like its historical equivalent?
Yeah, there certainly are groups who are highly motivated to put more fire on the ground to accomplish a variety of management objectives. There are groups specific, like my study focused largely on the Great Plains, so we can talk about that. There are groups interested in protecting, and fire is actually going to be a critical tool to conserve grasslands in the future because of ongoing woody plant expansion. It's just managers are having a harder and harder time keeping up with expanding woody plants like eastern red cedar, and fire is an important tool to manage that. So yes, there are plans and there are people actively implementing what's called patch burn grazing, which is a management strategy where a pasture is burned that takes advantage of this pyrrhic herbivory interaction where herbivores go to recently burned patches and preferentially graze them. So it has an application in kind of a livestock production context, and it has this added benefit of controlling woody plants from a livestock production standpoint, which is a predominant land use in the Great Plains today. If you want to continue to produce grass, there needs to be a strategy for dealing with expanding woody plants because forage production goes down by quite a bit and it decreases rapidly with woody plant expansion. So yeah, I think what you see in the way that fire is being applied, it's not being applied to the same extent that it would have been historically. The areas are smaller. The burning is still less frequent often. So it's not a true like historical analog, or it's not truly restoring past fire dynamics. But and also there's these attempts are limited in scope, like the landscapes are more fragmented, you know, so it's harder to burn and it's harder to burn larger areas.
Our next question is very interesting talk. Are the biomarkers you use resistant to biogenesis on these timescales, or is it something you have to take into account?
Yeah. So on a 2,000-year time scale, there's certainly they can be potentially degraded in a system like Buffalo Ford Lake, where we have less concern about post-deposition changes, just because it's a deep, cool, dark lake that hasn't dried out over that time period. If we're looking at a wetland that's periodically wet and dry, then we'd probably have some problems. There's like no reason to think that they break down, as some intrinsic property, because there are these steroid compounds, similar ones. These steroids are indicative of animal life, and they've been linked to the earliest fossil animals. So not even tens of millions of years, but longer. So they can persist for quite some time. So yeah, they can be degraded. And in this setting, we're not so concerned about post deposition degradation, though.
Our next question is, why were the bison not impacted by the megafauna extinction? Are bison not also megafauna by some metrics?
Yeah, I would say the bison would qualify by many metrics. I would say that they were impacted, but in a different way, right? So this is suggestive to me of potentially ecological release, while other larger herbivores that were consuming a great share of biomass were going extinct. Bison may have been able to take advantage of increased availability of forage, of high quality forage. This is also a time when bison body size was reduced dramatically. So I wonder about whether a change from inter-specific competition to intra-specific competition may have influenced what kinds of, basically what genotypes were selected for, or what phenotypes. So we have bison, we have other megafauna going extinct. We have bison expanding, at least during part of this time. We have bison body size decreasing, and bison, as the distribution data show, during the Younger Dryas, which is during this extinction window, bison are widely dispersed. So perhaps bison benefited from megafauna extinction. Perhaps they even some people have hypothesized that they may have even contributed somewhat because of competition. So yeah, it's a really, I think bison are a really interesting key to understanding megafauna extinction in North America.
And our last question from the audience is, are there also fecal biomarkers for carnivores such as wolves for the Yellowstone study? Has this been studied?
Yeah, so the example I gave had dogs. The study by Heralt et al. They had dogs in here. Humans are carnivores too. Like, so we have a meat component in many of our diets. So yes, they've been studied in carnivores. You can expect to see far more caprostanol in carnivores and far less plant steroid derived compounds like the ethyl caprostanol. So yes, but not, I mean, to answer your question, it has not been studied to my satisfaction, let's just say. I am not aware of studies that have explicitly tried to reconstruct past carnivores with specific biomarkers in a wild setting and not with a lake sediment. So I think there's some potential there with bile acids especially, and it's an area that I am really motivated to work on more. So happy to talk ideas with anybody if they have any thoughts on that.
All right. Thank you so much. So I'll take the screen share from you. Thank you everyone for joining us today. Please fill out our weekly feedback form. It's up in the chat box. And do join us next week. So next week we will be joined by Victor Rademacher, who will talk to us about ornithischian dinosaur evolution. So we have tea time with our speaker today, starting in a few minutes. So it's just an informal conversation where we ask questions to our host and have a little chit chat. So before we start today's tea time, we have a question for the week, which is, if you could be one animal, what would you be and why? And now think about that while we go into our tea time session. So we resume after a 2-minute break. See you all again.